Hi guys, Dr. Lando here with a video on cellular anatomy. So we're going to look at a plant cell. I have 2 pages here on my video. The first page is plant cell, second page is animal cell. So we'll toggle between them as we draw the structures and talk about what is in each one. Okay, so both plant cells and animal cells are eukaryotic cells, so they are going to have the cell membrane, nucleus, organelles, much more complex structure than the prokaryotic cells that we looked at. So we're going to start with the plant cell. First thing we're going to talk about is the cell wall. Okay, so we're going to draw this guy kind of big. And the cell wall is thick. It is usually drawn and seen as being kind of a greenish color. But it is thick, and it is made up of cellulose. Cellulose is a polysaccharide. You recall, it is indigestible to us because of the way the glucoses are attached. Remember that every other one is flipped. I'm drawing it nice and thick here to show the thickness of the cell wall. So what's the purpose of the cell wall? It gives the plant support, plant cell support and structure. Okay. I say the cell wall is like a thin cardboard box, so we'll come back to that thought, but let's label this. Look at it. So that's the cell wall, and I will not be going over to the animal cell to draw cell wall because animal cells do not have cell walls. Only in this demonstration we're doing, plant cells have them. Okay. Other eukaryotic cells do have them. Protists and fungi can have cell walls, but animal cells never ever. Okay. Inside the plant cell wall, you will find the plasma membrane, and this plasma membrane will look just like we drew and talked about in class: phospholipids, cholesterol, membrane proteins. Okay. So cell membrane, or you could call it plasma membrane, plasmalemma. So let's go over here to our animal cell. And with the animal cell, I'm going to not draw him as a square. I'm going to draw him kind of as a amorphous shape there. So that's our cell membrane. Down there. Cell membrane. Okay. All right. Next up is the nucleus. And the nucleus, we're going to draw in a blue color. Okay. The nucleus is the headquarters of the cell. We're going to draw it over to the side here on this plant cell wall, and our plant cell. The nucleus is where we find the DNA. So I'm going to draw the DNA here. I'm going to draw it like X's. Okay, it's not how it always exists. When we draw it like that, it usually means we're talking about the cell getting ready to either copy its DNA or split its DNA, right? And so this is our DNA. This whole thing is the nucleus. Okay. The DNA in that form, we would call those chromosomes. Okay. All right. So that's where the, as I call it, the library is housed. That's all of the genes needed to make this plant cell. Okay. To make the whole plant. Okay. Now the nucleus, I only drew a single ring around it of membrane. Anytime I draw one of these lines here, we're talking about membranes, but it really is a double phospholipid bilayer. So that's unique, double phospholipid bilayer. And there are holes. I need a smaller That's as thin as I get for a eraser. Oop. Undo that. Okay. No, that's okay. So it has holes in the membrane. Okay. So let's scooch back there. Let's label all of that. We've got our nuclear envelope. Which is the nuclear membrane. Okay, we've got the nuclear pore. Okay, allows things to go, proper things, to go in and out of the nucleus. Okay, remember that the nuclear membrane is special because it is a double PL phospholipid bilayer. That's special. Okay. All right. So that's our nucleus. Inside of the nucleus, like I said, you find the chromosomes. Let's go over to our animal cell and draw our nucleus. Again, we're going to draw a circle. I'm going to draw another circle because this is a double phospholipid bilayer. I'm going to give some nuclear pores there. Okay. And then we're going to put some chromatin in it. So instead of drawing the X's, I'm going to just kind of draw it all scattered about like this, kind of mottled in its appearance is a good description, like that. And most of the time when you look at a cell, this is what the nucleus is going to look like. It's not going to be all organized into those nice little X's, okay? So we call this chromatin. Okay, and so this is all of the chromosomes, the DNA, and the proteins that are in there, and you can see them spread out like that. Okay, still have the nuclear envelope and the nuclear pores. They got really big on this one. My eraser was a little overzealous. Okay. Some other stuff you might find in the nucleus, you will find in the nucleus. The nucleolus. So draw a dark circle in the middle of the nucleus. I call it nucleolus. You'll also hear it as nucleolus. Okay, so the nucleolus is there. The nucleolus is an accumulation of RNA and proteins. Remember I said that the nucleolus is a factory. It's where we make ribosomes. So we're going to find ribosomes in there. You're going to find those ribosomes are going to be in various states of completion. So just like you would find in a car factory, you might find some completed cars. You're also going to find some not complete cars. I'm trying to draw those as little dots. Ribosomes are always going to be drawn as a little dot and a big dot. Small subunit, large subunit. Okay. All right, let's go put our nucleolus over here. We're going to give it a big purple dot. I'm going to not label everything on this animal cell just to save time on this fiddle. Okay, so I got some ribosomes in there. Okay, it'd be good for you to go through and see if you can pause it and identify the things in the animal cell that I don't label. Okay. All right. Move that over there. All right. So we got our nucleus complete here. Oh, one other thing, sorry about this. Mention that some of these ribosomes are sometimes on the surface of the nuclear membrane. Okay, so we'll draw some ribosomes out there. Do the same thing over here, a few ribosomes. I'm not drawing nearly enough, and they would be much more plentiful, but I just want to make sure to remind you that we find ribosomes in different places: nucleolus, nuclear envelope. Okay. All right. Also want to mention about the ribosomes that they are unique for eukaryotic cells, which is nice. They're different in bacteria, so we can target them with antibiotics. Okay. We will also find ribosomes out here in the cytoplasm. Okay. Some of them will be joined together to make large complexes, polyribosomes they're called. It's hard to draw a nice circle on here. Okay, so there's some ribosomes. Let's go draw some ribosomes free floating out here. Good. Okay. Good. All right, so let's go on to the membrane system. Inside of the cell, there's a very complex membrane system that allows for kind of a stepwise processing within the cell. Okay, so the first part of the membrane system that we're going to talk about is the endoplasmic reticulum. And the endoplasmic reticulum is contiguous, connects to the nuclear envelope. So we're going to draw, I'm going to draw it kind of on this guy over to the side a bit. It does get a little squished. Okay, so we're going to draw kind of some squigglies here. Like that. I'm going to go on a bit. Okay, so that's going to be our ER, endoplasmic reticulum. Now there's two types of endoplasmic reticulum that we talked about in class. Actually, there's more than that, but in these cells there's only two that we're talking about. The rough ER, and these yellow, little pink dots should tell you that what's on them? Ribosomes. Yep, ribosomes rough. So, okay, so we're going to make this guy the RER, rough endoplasmic reticulum. That section right there. Okay. Rough endoplasmic reticulum. And remember what I talked about in class with ribosomes. What is their function as a job? The ribosomes are the workbench for protein synthesis. Okay, so we got to have a bunch of, if we have, everywhere we've got ribosomes, you think protein synthesis. So what's the function of the rough ER? Protein synthesis. That's its major job. Okay, the next thing here is the smooth endoplasmic reticulum, SER. Smooth tells you there are no ribosomes on it. Function of the smooth endoplasmic reticulum is different. It has quite a few jobs, and depending on where it's at, those jobs may be more important in one cell than the other. So lipid synthesis, you want to make some phospholipids or assemble them. It's going to happen in the SER. It's going to make some triglycerides. It's going to happen in the SER. Detoxifying things like any kind of drugs or alcohol that or toxins that a cell might come across are going to be detoxified in the SER. Breakdown of glycogen happens in the SER as well. And then there are some specific jobs for the other types of endoplasmic reticulum that we talked about in class. Like, for example, that one that was called the sarcoplasmic reticulum. That one was going to be really important when you talk about muscles, because its job has nothing to do with lipids or protein synthesis. It's simply a storage site for calcium. Okay, I'm not going to draw that in here because this is not a muscle cell. This is a plant cell. Let's go look at our animal cell and draw in our ER again. We're going to draw the rough ER being continuous with the nuclear membrane. And then we're going to continue on to a little more down here. Okay. we're going to zoom in so we can get a nice picture. Draw some ribosomes. When I'm drawing these ribosomes, immediately your brain should be saying, that must be the rough ER, and that ribosomes, because they are the workbenches for protein synthesis, this whole guy must be responsible for protein synthesis, right? So that's our rough ER, and then this stuff here that does not have ribosomes is our SER, smooth endoplasmic reticulum and rough endoplasmic reticulum. All right. Okay, so on to the next thing here. So once we go through the SER, we're going to come to this flattened stack of pancakes, as I described it, right? What is that? That's our Golgi. And you'll hear this referred to as Golgi, Golgi body, Golgi apparatus, Golgi complex. I'm going to label them Golgi because it's easy. And our Golgi, remember, that's our post office, or UPS, whichever you prefer. They're going to package and ship products made in the SER and RER. So as products are made, and I will draw this again, maybe in a separate video or this one, we'll see how long this goes. As we make proteins or lipids, eventually they are packaged and shipped to where they need to go. That's the Golgi's job. It also can modify, as can the SER and RER. It can modify proteins, so cleave them or cut them or add sugars to them. Modification is a big job in this area as well. Okay, let's draw in our Golgi. Looks like a flattened stack of pancakes, and there's always little vesicles budding off of it. That's what I drew in the other one too. Vesicles are next. This is Golgi. Okay. There we go. All right, so what's coming off of there? Vesicles. Okay, inside of those vesicles, if we could zoom in on these guys, we'd find little product that was made in the SER or the RER, modified in the Golgi. I'm going to find some stuff in it. Okay, same thing here. These vesicles will find some products in them. Okay. So a vesicle is a membrane-bound sac that contains something. Now there are different types of vesicles that we will talk about. So we'll go into those now. These happen to be transport vesicles because they made something and they're going to transport it to the in our little world here. These are being transported to the plasma membrane to be maybe excreted from the cell or used in the cell membrane or something like that. Okay, also in the cell we will find other types of very specific vesicles, membrane-bound sacs. Again, that's what a vesicle is. So I'm going to draw this one round here, like that. Let's zoom in on him, give him some little dots in here. It's going to be orange. This is a lysosome. Remember, lysis means to rupture or destroy. Lysosome, forgot to know there. Lysosome, and it is responsible for digestion. So this is like the cell's stomach. So it's got enzymes for digestion. So if we were to have an organelle that needed to be recycled, this lysosome would take care of that. If this is a little amoeba here, this guy, let's say he's an amoeba. So he's out, or it could even be maybe a white blood cell. Okay, whatever it is, it's a phagocyte. That means it's a large eater. It's going to go and ingest particles and bring them in. So here's a little critter that it's going to ingest, a little bacteria or something like that. It's going to bring it into the cell. Okay, just like that. brings out these little arms right here, wraps around it, and captures its dinner. Well, that's nice. His dinner's in this nice little food vacuole, which is a type of vesicle. Okay. Now all it needs to do is find itself a lysosome, add those two together, and then you have digested food. Some purple to show that he's all broke up. Okay? So better like that. There. So the food vacuole plus the lysosome means dinner, digested food, digested particles. Okay, so this food vacuole is, was brought in from the outside, but perhaps this didn't come from the outside. It could have came from within the cell, some broken down old organelle that needs to be destroyed. Same thing. That's how we would take care of that. Okay, so here's plants also, not necessarily bringing things in from the outside like bacteria and whatnot, but they also have to digest any old things that need to be broken down or clean up if there's an injury in an area and help clean up an area. Okay, peroxisomes. We'll find peroxisomes as well. What color should we go here with these? Let's make them this blue color. And there's no way for me to draw them differently. They are just little vesicles. I guess with the peroxisome I could draw, this is what I could do to make him look unique. We're going to put a little square in there. We're going to put a little square crystal inside of our peroxisomes. There. That way he looks unique. So let's go with that. And this is the peroxisome. Remember, the byproduct of chemical reactions can be H2O2, which is peroxide. We don't like that. It is a free radical, starts breaking down things that it shouldn't. So the peroxisome contains catalase. Okay, and that catalase is an enzyme that breaks down peroxide. Make sure that you know the formula for the breakdown of hydrogen peroxide. I'm not going to write in here, running out of time here. Okay, and so those are the three big things under vesicles, actually four big things under vesicles. Transport vesicles, lysosomes, peroxisomes, and food vacuoles. On here we could draw some peroxisomes to make sure that we are, what color did they give that guy? He's blue. Okay. And then he had a yellow crystal. That crystal that's in there is catalase. And the bigger the crystal, the older the peroxisome. Okay. All right. Okay. One other thing I should mention in the animal cell about the vacuoles, and I don't want to draw it into this guy here, but make sure that you check out contractile vacuole. Remember, that was found in our freshwater protists. It was used to remove extra water that comes into them so that they don't explode. So it's kind of like a sump pump for the for those critters, freshwater protists and microorganisms. Okay, and then the last big one. This is really the big important vacuole that I should mention is our central vacuole, and this is only going to be found in our plants. Okay, we're going to need a lot of blue here for this. Let's get a nice light color blue. Okay, so the central vacuole is big, and it fills the space between the organelles, and its function is to hold water. Remember, I said that the cell wall in a plant is really like a, oh darn, sorry about that, is really like a weak cardboard box. And so if you had a cardboard box and you tried to sit on it, it would collapse, right? But if you took that cardboard box and you put a let's say it's a really strong water balloon inside of it, really thick, and you filled it full of water, now that water balloon will reinforce the cardboard box, and now you can sit on the box. You sit carefully. So whoa, let's undo that. I need to get rid of my, there we go, over here. So we just drew a central vacuole, unique structure to find in plants here, and its purpose is to hold water for the plant. Its purpose is to increase turgor pressure. Capital P refers to pressure. Increase turgor pressure. And that's the pressure that water balloon, the central vacuole, exerts on the cell wall and cell membrane to keep it inflated. Remember, I talked about plants. If you don't water them, they wilt. And that's because the central vacuoles are dehydrated. And if you have certain plants will really wilt down, and you can water them and they just seem to come back to life. And that's because when you water them, the water goes back into the central vacuole. The central vacuole pushes against the cell wall and the plant stands back up. Central vacuole also has other jobs. It can store stuff, a plant can store stuff in the central vacuole. It can put things in there that are, for example, stinky, like sulfur, or things that are toxic. It could be in an effort to protect the plant from being eaten, or it could be a way that the plant just metabolizes things to survive. Okay. Also in the central vacuole, you're going to find ions, sugar and ions, that will draw water in, that act like sponges to suck water into the central vacuole and keep it there. Okay, so now the next organelle we're going to talk about again is going to be specific to our plants. I'm going to draw it as a green dot. I want a light green dot. Okay, and that is going to be the chloroplast. I'm going to draw them all around here. The chloroplast is a type of plastid. We talked about a couple of different types of plastids in class. Plastids usually contain a pigment, or they act as a storage site. In this case, the chloroplast does contain a pigment. It's called chlorophyll. I have room to write this, goodness. Okay, let's go to a thin one. So the chloroplast is a type of plastid, and it is containing chlorophyll. Chlorophyll is a pigment, and it is able to absorb sunlight, and it is the reason that the chloroplasts are responsible for making ATP, which is used to make sugar. Okay, or other biomolecules. So we say that plants are autotrophs. They can make their own food. How do they do it? They've got chloroplasts with chlorophyll, and that's going to be an entire chapter in this class. So I'm not going to go into too much detail here. I just need you to accept that fact right now, that chloroplasts do that. Okay. All right, we're not going to draw our chloroplasts over in our animal cell. Have them over there. Okay. All right. Next up is going to be mitochondria. Where in the world am I going to put these guys? Put them right here. Plant cells do have mitochondria. They are important. I'm going to just draw him in there, but we're going to go over to the animal cell because I've got more room over here. So the animal cell has a lot of mitochondria. And I want you guys to review that endosymbiont theory for mitochondria because I think it is so interesting and important. Make sure that you understand why we believe that mitochondria came from a primordial cell, essentially adopting a little bacterial cell. So what is a mitochondria? Mitochondria are and I know it's cliche, but every textbook says it and every teacher pretty much says it. It's the powerhouse of the cell. Ooh, hello. Okay. It's the powerhouse of the cell because it makes ATP. Super important. It makes ATP. Can you make ATP without a mitochondria? Absolutely. Can you make a lot of ATP without mitochondria? Not as much, right? So remember, with ATP, if you burn one 6-carbon sugar, you can get about 30 ATP, whereas if you don't have the mitochondria, you're only going to get 2. So this was a big upgrade for eukaryotic cells to have mitochondria. Now they can extract so much more energy from one sugar molecule. Okay. And again, we have a whole chapter on that. That's a fun chapter on how ATP is produced. Okay. All right, last thing we need to talk about is the cytoskeleton. And this is not going to be something that I can draw in really easily here, but maybe we'll just do, I'll go down here and just write cytoskeleton. So we know that cells don't have bones and plants don't have bones, so we have to have some kind of internal structure, and that's what the cytoskeleton is. It's made up of proteins, because everything's proteins, right? Made up of proteins, and those proteins can be thin, medium, or thick. So we've got microfilaments, intermediate, and we've got microtubules. Okay. The microfilaments are going to be the thin ones. Intermediate, that's pretty obvious, they're medium sized, and the microtubules, these are thicker. And so depending on what you're talking about in biology, you will come across these terms. So it'll talk about microfilaments when you get to the muscle chapter, and microtubules I'll mention right now when we talk about cilia and flagella. Okay, and the intermediate fibers are going to help with our attachments that we talk about later. Okay, with cilia and flagella, I'm going to go ahead and draw these on the animal cell. Remember, cilia are short, hair-like projections. These are covered with plasma membrane, and their job is to, if it's all around a cell, it could be for moving the cell itself, but inside of our body, cilia's job is usually to move a fluid or a substance, to move fluid or substance. Recall the mucociliary escalator that I drew in class showing the trachea and how it was moving the mucus up to the pharynx to be coughed out. Okay, that's cilia. Sometimes you'll have flagella on a cell, like a sperm in humans, and the flagella are longer, fewer in number, only going to have 1 to 2 of those. And these are for cell movement. Has a whip-like action. Okay, and for these structures, a common thing that you will see, and you do see it in your textbook, they will say that these have a 9+2 arrangement, and that is referring to if you were to cut the flagella and look at it end on, and I will not be able to draw this really well, but there will be 9 pairs. 3, 4, 6, 7, 8, 9 pairs of microtubules all the way around the outside, and then there would be 2 in the middle. So 9 pairs of microtubules plus 2 more. Okay, and that is unique. Why they make a big deal about it? Because that's a eukaryotic flagella. It's different than a prokaryotic flagella. Okay. And so some, obviously sperm are the only flagellated cells in humans, but other little critters like protists will have flagella so that they can move along. All right, I'm going to stop there. This has turned out to be a really long one. So if you have any questions, please let me know, and realize that this is an overview. If you go back through your notes that we talked about in class, you know you will find a lot more detail on what each of these individual organelles and structures do, and you are responsible for that. So go ahead and study that. If you have any questions, give me a call.